So tonight we have Brandi, Kiel, Reese with Outwit, Outlast, Outplay, a game of bacterial survival in the deep ocean. And a week, or I'm sorry, 2 weeks from yesterday, we have The Maid of the Mist Goes Electric, which is talking about the boats at Niagara Falls have been replaced with a fully electric fleet. And they're going to talk about some of the reasons behind that and how it's going. Last year was the first year with those in the river. And those are the boats that go up to the base of Niagara Falls. On April 14th, we'll have Dr. Vandana Singh. And I don't know her title yet, but she is a very, very interesting professor of physics and science fiction author with lots of interesting thoughts on how to teach climate change. And on April 27th, we'll have our Earth Day program with Jay Burney. On May 12th, we'll have Annie Fresh Ozar talking about the science of Muppetry. She is a four-time Daytime Emmy winner for her work on Sesame Street. And then on May 25th, we'll have a climate change education panel. And that'll be our final session for the spring. And opening in April, we'll have Six-Legged Science at the Museum of the Earth, which is one of the public venues of the Paleontological Research Institution. And I am about to hand it off to Brandi here. And I will note that I met Brandi when I sailed on the Joydes Resolution, which I expect she'll say some about tonight. And I'm going to hand it off to Brandi and stop sharing my screen so she can start. And welcome one and all.
Thank you very much. Let's get this started. Hang on, just had it. All right, can you see the presentation? Okay.
Yes, thank you.
Yes. Great. As I was saying, or as John was introducing, yes, we did meet on the Joydes Resolution. You can see that in the bottom right. That's the image of the ship, and I'll talk more about that. And as you can see, it's a drilling ship, but we're not going to be talking about oil drilling in any way. This is definitely focused on research and understanding, what is deep within the earth, within the ocean. I'm a microbial ecologist. That means I study microbes, bacteria, archaea, and even fungi, the occasional fungi that live in the deep ocean, deep subsurface. So that is what is below the seafloor. I'm at Dauphin Island Sea Lab, and I'm also at University of South Alabama. Dauphin Island Sea Lab is a marine station, and I'm also appointed at South Alabama as well. Oops, let's see. There we go. I think we're on track. I like to start off with giving some thanks, because that's what's most important to me, is the team that puts these, makes this research possible. And so I couldn't do that without my lab group that I've got. And then I also have, of course, IODP to thank. That's the International Ocean Discovery Program, and funding sources from NSF, from NASA, and the American Chemical Society, it's to name a few, as well as the Sloan Deep Carbon Observatory, Sloan Foundation Deep Carbon Observatory, and then NSF's Center for Dark Energy Biosphere Investigation, CDEBI. So that's a mouthful, but all with the trying to understand, what life is like in the subsurface. And this is what we're talking about. This is a cross-section of the ocean, and of course, the what's below the ocean into the subsea floor. We have areas, that we're very used to going just right off your continental shelf, going out in a small boat and collecting water, collecting samples, and staying within that photic zone. That is what history has taught us to do. But what we try to do is go further, go deeper, and try to explore areas like in the deep sediment or even into the ocean crust. And from our perspective, from my perspective particularly, there's some interesting chemistry, there's some interesting physics, but there's also very interesting microbial life, bacteria and archaea that live deep within the earth and thrive, not only just are surviving, but also thriving. So I'm going to introduce you to the program through a short introductory video, and then we'll talk a little bit more about how we get these samples and then what these samples mean.
Just off the coast of Newfoundland, scientists aboard the JOIDES Resolution recovered sediments from a depth of 1,000 feet below the seafloor. They pulled up a core with a strange layer, a dark band of ash and rare metals. This layer is 66 million years old, the same age as the youngest dinosaur fossils we find on land. Dinosaurs roamed the earth for over 150 million years before this time. Then suddenly, they disappeared. An asteroid hurtling through space hit a shallow ocean off the coast of Mexico and vaporized upon impact, unleashing massive fires and volcanic eruptions. Dust and ash filled the atmosphere, blocking sunlight for many years of bitter cold and darkness, wiping out 3/4 of Earth's plants and animals. We know about this natural disaster because it left its mark right here in this core and others found around the world. Thankfully, some creatures, similar to the ones we know today, survived this catastrophic event, and the Earth rebounded, making way for new species to develop and flourish, like our own. The ocean holds a most impressive record of Earth's history of over 200 million years. Not in its waters, but in the seafloor beneath. Particles of sand, clays, ash, and the remains of living things collect here over time. Younger material building up on top of older. Earth scientists set out on specialized drill ships that, like time machines, allow them to travel back to periods when palm trees grew on Antarctica or to an Earth mostly covered in snow. They drill deep into the seafloor through layers of time and extract cylinders of mud or rock, cores that are carefully pulled up on board and delivered to labs on the ship. Here, scientists become detectives of Earth's past, using clues from the fossils of single-celled creatures called plankton to decipher what the Earth was like thousands or even millions of years ago. Like this core, drilled in the North Atlantic, it contains species of plankton that thrived in the freezing waters of the last ice age, a time when ice sheets covered large areas of land in the Northern Hemisphere, 24,000 years in the past. Ocean drilling is dedicated to pulling cores out of the seafloor, but in some cases, we actually put something else in. After pulling the blueprints, these scientists placed a string of instruments into the drill holes. They measure any slight shifts below the seafloor, shifts that could lead to an earthquake deep inside the Earth. This is valuable, especially in Japan, where some of the world's deadliest deep-sea earthquakes are born, triggering massive tsunami waves that devastate coastlines all around the Pacific Ocean. Every tremor along this fault is now recorded in real time. giving completely new insights to the buildup towards large earthquakes, where they start, and how their energy spreads throughout the Earth. And with a network of instruments in place, we can now detect tsunami waves earlier than before, helping more people to safety in time.
Well, this scientific drilling it has a long history, and you may not, this may be your first time to hear about it, but it's actually has been going since 1958, almost continuously. It started with Project Molehole, which was an attempt in the early 1960s to drill through the Earth's crust to get samples of the Markovitch discontinuity, which is that boundary between the Earth's crust and the mantle. So you've got your sediment, then you have your the crust, and then the mantle. And the project was intended to provide, kind of an earth science complementary to the space race. And that, over the years, this program had very important discoveries that have been made to help us understand subsea floor processes like plate tectonics, subduction zones, and how hotspots form. And then, of course, climate change and sea level rise, as we just talked about. And then also, microbial life that has been, an understanding that has, or broadened our understanding only in the past 10 years, about 15 years since, we've really tried to, grasp, the extent of microbial life. And as you can see, it's been a continuous program under various names. Each time it has been refunded by the National Science Foundation and its partners. It's a very international group. So it's not just NSF, but also partners in Europe and, of course, Japan as well, but many other countries that have participated and supported both financially and scientifically in the program. And so I mentioned the JOIDES resolution before, and that's operated by the United States out of Texas A&M College Station. But there's other platforms that we rely on as a program cooperatively. And so the Japanese own the CHICU. It's a different platform that can go in different waters. And then mission-specific platforms that can go into shallow waters and maybe into, that is more ice covered, or can also be, like I said, into just areas that are more maybe specific or difficult to get into. And of course, sampling is always a team effort on board. It's a very international science party, very interdisciplinary science party, from geologists to chemists to, of course, physicists, paleontologists, thank you, and of course microbiologists as well. And this was some fellow microbiologists that we were sampling, from the interior of the core to assure that it stays sterile. And that's another part that we're going to talk about today, is the sterility of this. And then, of course, maintaining anoxic conditions and characterizing these cores as they come up. And archiving is a very important part of this process as well. And so we've had, thousands of holes drilled over time, hundreds of expeditions. You can still see that there are, definitely some areas that we could still pursue. This figure is a couple of years old, so there might be a couple new spots that we haven't haven't included on here yet, or that haven't included the updated figure on here yet. But these are just the ones that were focused on microbiology, the ones in these yellow stars. And in fact, this one right here is actually setting sail April 1st. I don't know if you can see my pointer. There it is, that you can see on April 1st. So that's a little bit, it was rescheduled from 2020 to now 2022. And so we're I have a student going out on this expedition, and we're just, I'm going ahead and putting that star on there because I think it's going to happen. It's, we've got 2 weeks before that one sets sail. And then these are the only other sites that have been, focused on microbiology. And so that just tells you, and the reason why I mentioned this is that we still have a long way to go to understand life below the seafloor. And so what do we know about microbes? Well, it has been all over the news, most particularly, viruses, but also, bacteria and archaea. Those can't be neglected as well. But what we know about microbes is that they are, pathogenic, they're infectious, they're causing disease or even the, bad breath. But there's a lot of good that comes from microbial. In fact, they're much more good than harm. Of course, we use it in our natural fauna within our bodies and in digestion. Of course, nutrient cycling and carbon cycling. Hugely important. And so our view of life is, what's warm, what's, where the light touches, what's oxic. In fact, you talk to most, most people, and that is how we see our biosphere. But the Earth's view of life is something a little bit more cold, of course, dark, suboxic to even anoxic. And this is, if you think about 70% of the Earth being covered in, with ocean, and of course, below that ocean being a landscape of subsea floor and extending kilometers into the Earth. And of course, the crust below that subsea floor, the vast majority of Earth sees life in this way. I really love this slide, or love this image, I should say, with it's a drop of seawater there on the left, and that's what it would look like under a microscope with cells that are lit up with a stain. It's just a DAPI stain, and it reminds me of stars in our universe. There are about 1 to 10 million bacteria in 1 million to 10 million bacteria in a drop of seawater. And that's about a milliliter of seawater. And of course, viruses too. The big dots that you see here is our bacteria, but those little tiny dots, those are viruses. And there are about 10 to 20 number of viruses as bacteria. So for every bacteria or archaea that you'll have about 10 to 20 times as many viruses. And again, they're beneficial and important, definitely not pathogenic. And compare that to the number of stars in our galaxy, our universe. There's a common belief that about 99%, and that of course is changing somewhat, of the environmental microbes can't be cultured. That means they can't grow under laboratory conditions. That is like I'm showing here on a plate. They can't, we can't replicate everything, every possible condition in the lab or in a bottle that they experience in the environment. So what do we do? How do we tackle that problem if we can't grow them? Well, the objective here is to, try to get their DNA. This actually, this figure was from 1997, in which that was really when we started to move from the 5 kingdoms to 3 domains of life: bacteria, archaea, and eukaryotes. I like this particular one, and I'm going to show you another tree of life in just a moment, but because it really shows, our related it, the diversity, I should say, of the bacteria and archaea, which we call prokaryotes. Here you are with Homo sapiens. Zea is corn. Croponus is slime mold. So you are more closely related to slime mold or to corn, genetically speaking, than one bacteria is to another. And certainly bacteria to archaea, both single-celled organisms, vastly different as well. Here is a new tree of life, though, published in 2016, which you may or may not have seen, and really using genomes. That means all of your DNA, and comparing, this is comparative genomics, comparing all of the DNA to that has been available within the databases collected so far. And you can see those in red. These have no cultures, right? So this is just the DNA that's been extracted from whatever particular organism, and using the whole genome, the whole complete genome, to create this tree of life. And so here's bacteria here at the top. Here are your archaea here towards there to the left, this branch, and then that branch coming right off of that archaea, there's our eukaryotes. And there you are, somewhere in there. So eukaryotes are more related to archaea, and then bacteria more distantly related. This, as earth microbiologists, or we call ourselves also geomicrobiologists, we ask questions about where on Earth we can find the microbes. We ask questions like what limits to life are, for example, how hot or how cold or how deep life can exist. The deeper you go into the Earth, there are less yummy nutrients and carbon for bacteria and archaea to munch on. So the depth is also an extreme environment, as much as cold or hot would be. So you take a look at this picture, and this is a cross section again of the Earth. And at the top, you may have 10,000 cells in a gram of sediment up here. Let's see, right up here. There's my mouse. There we go. About 10,000 cells per gram of sediment. That's a little, maybe it's about a quarter of a teaspoon or so, maybe a little between an eighth and a quarter. And then, as you go deeper within the Earth, you can get as deep as 1,500 or 2,000 meters, and there you only have about 10 to 100 cells in a gram of sediment. And so it's pretty deep to find life, but this life isn't like bacteria and archaea cells that we're used to interacting with in the surface world. These microbes are existing in a very harsh environment and are often dormant, meaning that they're just not active. They're not, maybe we're just going to find their DNA, and we can't find, the other thing that I'm after is the RNA, which is a proxy for activity. And so we kind of call these guys zombie microbes, because they are often barely even alive. They're just, but they are intact. Their DNA, is present. It's whole, and what they're doing is something that we're always trying to ask ourselves. So in addition to asking how deep they are, and also where they are on the globe, as in biogeography, we also like to ask how many microbes are on our planet, particularly in the deep biosphere. So we've worked for a few decades to survey the abundance of life below the seafloor, and the estimates have been refined over the past decades as we get more data. But of course, thanks to programs like IADP that enables to drill deep into the seafloor. The corollary to that in the continental world is the International Continental Drilling Program, ICDP. So that's another way to answer these questions. And estimates suggest that 10 to 30% of the biomass on the planet may be beneath the seafloor. And that's a wide range, 10% to 30%. And you ask, why would there be such a wide range? Well, and the question is, where is our data coming from? A lot of our data comes from these coastal, samples. They're in black, which are going to be higher biomass, more cells. The cells are going to be bigger, and the cells are going to be more numerous. And that's because there's more nutrients to munch on. We have a lot of nutrients that flow from the continents, meaning your nitrate, your phosphate, carbon. And so that's going to feed your microbial life. And as you get out here into the deep ocean, just fewer and fewer cells. And so it's really, every time we go out, we get more information and we can refine these numbers and refine these estimates. And you can see there's a lot of holes to be filled. I like this, that there are 10 to the 29th cells globally in the marine sediments, and 10 to the 21st stars in the observable universe. So the number of the cells in the subsurface also, to put that into perspective, equals the number of cells in the ocean as well. So the water column equals what is in the sediment. And that represents just a fraction of the total Earth biome that we know about. One caveat to this is all the cells that were counted, and you saw that first picture that had those, the green dots under the microscope, that's a DAPI stained bacteria and archaea. That's only bacteria and archaea that are counted and using the methods used. So only sediment was also counted. So meaning that there's also marine viruses and marine fungi that also add to that biome. I know my viruses aren't technically life, but they do add to that, contribute to that biome. And only sediment was counted as well. And I'll talk about the crust. Like I said before, what's below the sediment into the crust is also equally as important. So are there microbes below the sediments? Well, of course. And this is one of the things that is much more recent development. And we use those observatories to try to understand that. And that is what I'm going to show you here. It's called a circulation aviation retrofit kit.
What is a cork? Of course, it's something that plugs up and seals an opening. But if you're interested in what goes on deep inside the ocean crust, your cork is at the bottom of the Pacific Ocean, more than 1.5 miles down, on the Juan de Fuca Ridge. It's sealing a borehole drilled 20 football fields deeper than the bottom into the seafloor. These corks are hubs for scientific research. They're subseafloor observatories. The upper ocean crust is the largest aquifer on Earth, holding as much water as all the ice caps and glaciers on the planet. This is mostly salt water, not fresh, but like fresh water on land, the salt water below the seafloor is in motion, moving from place to place across vast distances. Corks let people explore this undiscovered realm of water, rock, heat, and life inside the ocean crust. These corks on the seafloor are systems of hardware, plumbing, seals, and scientific instruments. Corks allow scientists to measure and sample pressures, temperatures, fluids, and microbial life along the boreholes. The data they collect can help them understand how microorganisms may have developed on Earth and how they could have developed on other planets, how the hydrologic cycle extends from continents to the seafloor, and how carbon is transported and might be stored within deep reservoirs, which could have a significant impact on global climate. Installing the corks was no easy task. It required the power and capability of the research ship Joydes Resolution to drill the holes. Once the core and the drill were removed, a re-entry cone, a funnel, guided the support pipe and instruments down the study hole. Then it was sealed from the ocean, corked. After letting conditions settle and return to normal, scientists began taking samples. The top of the cork that sticks out from the bottom is called the wellhead. It's surrounded by a round ROV platform. The area above the platform is divided into bays. Each bay contains the ends of long tubes that run from the wellhead down through the seafloor seal and deep into the borehole. These tubes connect to instruments and sampling points at different depths, allowing researchers to access these intervals over time to collect data and samples. The pressure bay contains highly sensitive gauges, batteries, and a computer that takes measurements. The long-term fluid sampling bay contains samplers that collect fluids from the depths. Another bay contains sampling valves for collection of microbial material from the crust below. And in some corks, there's a large ball valve used for a free flow experiment. Long-term sub-seafloor observatories are transforming our understanding of what it's like deep below the bottom of the ocean. Through monitoring and experiments, Corks are opening a window into a vast and hidden realm of crustal rocks, flowing fluids, and life.
So Corks have really allowed us access to samples that we haven't been able to access before. So of course the sediment was one, and the rock itself is another one. But then being able to put in those subseafloor observatories and accessing the fluids that are within the rock as well is really opened up, another, realm of understanding. And so one of my students just recently finished, and so I wanted to kind of highlight some of her work, and it's just been really, I'm really thankful for, her collaboration in this, because she's really brought an interesting biochemistry perspective, to the research. And a lot of the work that I'd been doing up until this point was, focused on panel A. So we have different mechanisms that we liked the, Outwit, Outlast, Outplay, and you can really think of it as a game of Survivor, in that, perhaps Outwit is, adapting to your surroundings. And I'm showing this because, you can have microbes that are adapted to, of course, oxygen-rich conditions. And these are our, could be our autotrophs. And then as we, move down this into a more anoxic environment within the subseafloor, we can have, microbes that are adapted to reducing sulfate to hydrogen sulfide, or sulfate reducers, or methane that's being reduced to, or that's being generated from the reduction of CO2 or hydrogen. And so we have, different communities of microorganisms, bacteria and archaea as well, that are adapted to, certain conditions. And then, of course, Outlast, dormancy. Dormancy is an important survival mechanism, and I mentioned that a little bit earlier, in that, an active cell is actively reproducing. It's growing in terms of the number of cells in its community and eventual, mortality. But then your dormant cell, is one that has, basically, shut off, all of its, necessary functions. Maybe it's just its housekeeping genes. Its housekeeping genes are just those that are, like DNA repair, cell wall repair, things that just keep your cell alive. And then what happens, to cause it to go into dormancy and then also come out of dormancy are questions, as we look at some of the genetics of a microbe's resilience. And then thirdly, in panel C, that Outwit, Outlast, Outplay, and being able to outcompete your neighbors. So if you can't adapt and you can't shut down and go to sleep, then why not just, kill or lyse your fellow cells, or at least deactivate them. So there's a bit of a, I guess, conundrum going on. So you can, if you produce an antibiotic, and antibiotic is just naturally produced by a bacteria, and it can kill, a strain that is sensitive to that antibiotic. But a sensitive strain to the antibiotic can also have a antibiotic resistance. And so all of that, can be resistant to the antibiotic, but it also doesn't incur costs. When I mean cost, I mean the energetic cost of producing the antibiotic. So it's a delicate balance. And when you're talking about, low biomass environments, the lysing of a fellow cell not only gives you access to those nutrients or carbon that you didn't have access to, but the cell itself is also a valuable resource with carbon, nitrogen within that DNA, phosphorus, sulfur, and hydrogen. So all of that gives that cell the a mechanism to survive. So we extract from cores a few different ways. The images that I have here are, we do some, and this is just a dummy core, because I wouldn't allow pictures to come into such a sensitive, to our sensitive cores. So we do this under, it's cryogenically frozen, and we can drill out the core and collect some samples while it's still frozen. And then also long before COVID was made, masks in vogue. We've been wearing masks in our lab. And so I think we were ready for the pandemic a full decade before the pandemic hit. So wearing that, we were not strangers to that. And the reason why we do that is we're concerned about, not about the samples, harming us in any way, but us harming the samples. To put it in perspective, your hands, even after you wash them thoroughly with, your antibacterial soap and saying happy birthday twice, you're still carrying, bacteria on your hands, 10 to the 5th, 10 to the 6th, bacterial cells, per square centimeter. So that's much greater than what I was describing when we're talking about 10 to the 3rd, maybe even 10 to the 4th cells per gram of sediment. And of course, your mouth is much more abundant in cells, than even your hands. So we've been concerned about, we don't want to sequence the graduate students. We'd rather, sequence the what cells that are, in our samples. So I have two ways to attack. We either isolate DNA and RNA and sequence it. And then we've got some what we call bioinformatics to analyze those genetics. And then another way is that we try to culture or cultivate by putting it through a fluorescence activated cell sorter. So rather than trying to grow it on a plate or in a bottle, we put it through a cell sorter, and we put it into, only the cells, the single cells into designed media or into treatments. And then of course, both result in comparative genomics. And here's some images of some what microbes look like in that, below the seafloor when it's stained with that fluorescent dye and viewed under the microscope. And a lot of these live in veins within the rock or within the sediment. This is some of that, the genetics that have come out of some of the studies that we've been doing. We've been trying to expand this globally. So out of the, I've been associated with 7 IODP expeditions, either, shipboard or shore-based science party, but collecting, cores from and sediments from various expeditions. And one of the things that we started to do as we amassed all of this genetic information, all this DNA, is let's pull out, how expansive these antimicrobial genes are. Where are they located, globally? One of the most important antimicrobial, it's antibacterial as well, gene is polyketide synthase, PKS. And so if we can take the whole, piece of it, it's about 25,000 base pairs. And we started comparing it to what was known. So what you're seeing here in blue, those blue dots here on the right, those are samples from what we call the deep biosphere, that is, below the seafloor. The ones that don't have blue dots, that they're plain named. Those are what are known antibacterial classes. Okay, so known antibiotics that are antimicrobial, not antibiotics, it's not pharmaceutical, but antimicrobial genes that are known, in our current databases. So we compared our unknowns here in the blue to our knowns. And one thing that started to come out from this is that our deep subsurface is just grouping together into its own clade. A clade is just this, a group of within this tree. And then, of course, our knowns that are most commonly derived from soils or, the surface world are distinctly different than those from the subsurface world. And that makes sense because they're evolving independently from each other. And these classes of the bacteria, this is within the continental and marine. Again, I said this was a kind of a biogeographic approach. So this is, the various classes of bacteria, and then there's archaea up here as well, up at the top, but that contain, the antibiotics. So it's been quite interesting as we kind of have been exploring these data. Another interesting finding, and this came from, so the other one was a global study. This is from the South Pacific Gyre, and this was a fungi that was found in, that we were able to both get the DNA out and also cultivate this from 176 million year old sediment. And that's came from this site right in the middle of the Pacific Ocean, within the Pacific Gyre. And we were able to name it, characterize this. And how did that survive? Well, it survived on, the dormancy, that dormancy mechanism that I was just talking about. It was not an active fungi. This is showing its, whole hyphae, but rather it was in the form of these spores that was locked away within the sediment. And so something interesting about this fungi, and this will be my concluding slide, is that it adds to the carbon pool by degrading recalcitrant carbon. And what I mean by that is that difficult to break down carbon. In a comparison, just kind of an analogy is, glucose or sugar, is like your candy bars. That's what, kids or, bacteria, archaea will go for first. That's the sweet, juicy stuff. And then the recalcitrant carbon is things that are, the carbons that are more degraded. So you've got your labile carbon and your recalcitrant carbon, and that can be your like woody lignin material, but it can also be more of your, oils and things like that, or hydrocarbons. And so these, we did the, in order to name this, we of course did the genomics on it, and we are noticing that it can break down that recalcitrant carbon, that hard to, consume carbon, and then through its body and through its necromass, add to the carbon pool by, through its own life cycle, in which bacteria then can, munch on the necromass of the fungi as well, and get at that labile carbon again. So it's just been an interesting circle of life. And with that, I'm going to conclude so we can have some time for questions. And yeah, thank you for having me. It's been a great pleasure.
Thanks, Brandi. That was great. Just mind-blowing stuff. And if I was a little bit more on top of my game, I'd have us do our peer-reviewed questions, but I forgot to do that at the beginning of the session. So we'll just open it up for questions, and you can do that by using the raise hand feature, which is on the reactions button on your Zoom screen. And there is a question in the chat, really cool. The new subsurface antimicrobial genes, do you know the mechanisms they use?
Meaning, let me try to clarify this. Mechanisms as in,
Antimicrobial mechanism.
Oh, no, so these are antibiotic genes. So these, we can do, so a next step, and I'm going to try to answer it, and I hope I'm answering it correctly. So the next step is to, identify, and I think we've done it for a couple of them, and we can produce some predictions. For the ones that we are, that are closely related to known antibiotics. So what we have to do is we go by comparing the unknown to what's known, and there's a database that's available to for us to compare against. And so we could identify a handful of them. So we know that it's a PKS gene, or other complement is the NRPS, which is non-ribosomal polyketide synthetase. So but only some of them can we identify, the actual product that would be produced. And the others are, and meaning that they're 100% similar to that product. But the others are, 70, 60, 50% similar. And so it's not similar enough to say that this is the product, or the antimicrobial product, or the antibiotic, but that is it's unique enough to be on its own. So no, we don't know exactly the biochemical pathway or the mechanism, other than based on the genetics, that it is distinct, or if it's completely similar. And yes, 50% is very different. So with those, we just kind of put those in a bin, so to speak, and say these are kind of categorize these as these are antimicrobial, but they are quite different as well. So.
And Rob's got a hand up, and then Carolyn after that.
Okay. Do we know if typically the most closely related living species to the species that we're finding down there are other species in the same environment, or is there constant adaptation to these really deep environments from microbes living at the surface of the seafloor?
Oh, that's an excellent question. I think I know where you're going with this. And I'll kind of answer it a couple of ways. Life in the subsea floor is not only low biomass, but one thing I did neglect is it's very, very slow. Some estimates using protein racemization experiments estimate 1,000 to 10,000 year turnover time. So depending on the lab and the experiment, and where they studied, some have said 1,000, some have said close to 10,000 years. So meaning that the whole biomass of that particular environment will turn over, reproduce, or like replicate itself over that time period. So evolution is very, very slow in the subsurface. So and another way I'm going to kind of tackle that question is the differences between adaptation and evolution, which that's another tricky continuum of another kind of a little bit I don't know question or answer. But adaptation says that if it's all right, I'm going to go back a couple of slides just to kind of give the visual. There we go. All right, so adaptation in that some of these microorganisms can do a couple of functions. They carry the genes. Now you're not going to see a methanogen, in an oxic environment. Some of those are very, extreme, adaptations or, differences, but some can carry, genes that they can turn on and turn off, denitrification, or, sulfate reduction. They can do one or the other. And I'm not at the risk of anthropomorphizing, and making it sound like it's a choice, but, when that environment puts that kind of stress on that cell, they can switch over and start, from, for example, denitrification over to, sulfate reduction, and still, subsist. So that was what I would call, kind of adaptation. But over time, through generations of cells, and also through lateral gene transfer. That means when, maybe a virus will take a piece of DNA and move it over to another cell, or even one DNA will take in, or one cell will take in the DNA of another cell. Those are different ways to do lateral gene transfer. Then you can start having some evolution in which a particular gene has been changed in some way, maybe deleted, maybe inserted, maybe mutated in some way, and then it takes that on as being beneficial. So does that kind of answer your question?
Yes, I mean, you're right. There's an incredibly rich subject that could sort of go on and on, but let me just, I'm going to ask one follow on that's related that maybe I should have asked first, which is how many of the things living in these deep subsurface environments live only in those places obligately,
right?
As opposed to maybe they're also living at the surface, but they can survive in these deeper places and
so how many are, cosmopolitan and, ubiquitous? And I don't have the exact numbers, but I can say that a vast majority are ubiquitous, that you can find them in, maybe in the water column, you can find them in, soils, or you can find them in, sediments. What you typically find, in a human microbiome, or even animal microbiome, will be vastly different than what you're finding, in a environment like a, sediment or a soil. But it is the community itself is, very different because of its, there's a lot, but there is a lot of functional redundancy in that you'll see, same types of microbes in, of course, different environments. There are some that are, have been very unique to just, hydrothermal vents, or unique to just, the subsurface world. So I would say about, maybe 10% are just going to be found in the subsurface, or, and I'm also kind of including, some vents, of course, and then some, and it also varies when I'm talking about like species. As you go higher up in the, taxa into phyla level, you're going to find a lot more, ubiquity, but species level, which is much more specific, about 10% are very, very unique to the subsurface.
Thank you. That's actually fascinating, and a much lower number than I would have expected.
So when you go in, and of course that starts to change. So like the, the genus, and, so on and so forth, as you kind of move up that
Sure.
Column. So.
Okay, Carolyn.
Yeah, can you hear me?
Yep.
Is there, do you know, similar research being done under land?
Yeah, so the International Continental Drilling Program, ICDP, has a complementary to to what's being done in the ocean. And so, they've have projects that really, span the globe. So that's one way, through continental drilling. But another way is, using mines. We had a project a few years ago. We were at in South Dakota at the Sanford Underground Research Facility, SURF, that was in Leeds, South Dakota, and in the Black Hills of South Dakota. And so that was a former gold mine turned into a research facility. And that's been, amazing source of information. Other projects that, are in South Africa gold mine and so on and so forth. And let me see if I can kind of point to some. I don't think I have a global, but we did take, continental data as well as, marine data in kind of exploring the antimicrobial gene ubiquity question. And so those are, just some of those that, some of those sites. And so, drilling, but also mines and caves too have been great sources of information.
Yeah, I remember hearing about the stuff in the South African mines many years ago. Yeah. So
Desulfurudus is one that is just very specific to those gold mines. So Sanford Underground Research Facility, which is the former Home State gold mine, and South Africa gold mine, very interestingly have one organism called Desulfurudus that is unique to those two sites. And they're obviously South Africa and South Dakota, two very different locations, same organism.
That's really interesting.
Yeah, well, what I was thinking about was hydrofracking. They use stuff called biocide, and apparently I just heard about this conversationally. They get a lot of slime clogging up their filters, and it doesn't seem like anybody has looked into it.
Yeah, so there's a researcher by the name of Kelly Wrighton, and she's at University of Colorado Boulder, I believe. And she has been looking at the microbiome of this, of this fracking. And so that's some new research. So I think it's only been kind of coming out within the past, just very recently, a few years. And but you're right, it has definitely been changing. It's been that's a little bit harder to access. The sites that I've been describing are things that through coordinated programs have allowed researchers to come in, and it is a little bit more difficult to access when it comes to working with some of the oil companies, especially on these kinds of questions.
You'd think it would be easy to access because they have a hole there and they have slime.
Right, of course. Yeah, okay. I should say, rephrase that. It is easy to physically go up to it if allowed to. And some of that, they have their own researchers that work on those types of questions, and they want to keep that kind of information about their microbiome more private.
Yeah, okay, thanks very much.
Pat?
How does this map into the search for life in other places like Mars? Is this used by, by the rovers and so forth?
Yeah, absolutely. So there's yes, there's very much a synergy between deep ocean exploration and deep subsurface research, and of course life on other planets. And so NASA's very interested in this research, and of course a lot of our scientists, myself included, crossover between the two worlds through astrobiology and Earth geomicrobiology. But so a couple of ways is that, as we understand life on our planet and the evolution of life on our planet, and the different ways that microbes are able to survive, we can start to try to understand, perhaps the conditions and survival mechanisms on other planets. So we need to keep teasing apart, the genetics and how microbes are adapted or evolve, here on our own planet. And then also through instrumentation, we mentioned the Corks, the subseafloor observatories. That has actually, been there was a tool that is, that has been lowered down into the cork, and it was first tested in some of our own holes, or some of our own boreholes. And this tool is a deep UV spectrometer. And so, in other words, it looks into the UV range. You get your visible range and your UV range, and it scans into the deep UV range. Their carbon that is associated with life, cellular carbon, has a distinct wavelength or a distinct signature. And whereas carbon that is, organic, not associated with cellular carbon, or inorganic carbonates, for example, have a different, wavelength, a different signature. And so you can scan, the sides of your borehole for this life. And much as the same as that, you saw the cells, kind of lighting up. You can see, the cells on a rock or in a borehole. Well, this same tool is now on the Mars 2020 rover. So the rover that went up a little while ago, the one that's up there right now, and it's called Sherlock, SHERLOC, and that is the same scanner that is now, after having been beta tested at the Sanford Underground Research Facility, where I was, and of course on some of the expeditions for IODP, it's now up, and it's complemented to Watson. So Sherlock and Watson go together, and Watson is LIDAR, and then Sherlock is that deep UV scanner to look for life. And it's so those data have yet to be understood or analyzed or processed. So stay tuned, and hopefully we'll hear something about whether there's carbon in that deep UV range that could be associated with a cell.
Yeah, and I'll just note that I have served on the U.S. Advisory Committee for this program, and there's a NASA representative on that committee for exactly these reasons. And I'm not sure if Carolyn has her hand up again or if it's just still up. I think it's just still up. Are there other questions? Well, if not, I will put a couple of links in the chat. One to donate to help support Science in the Virtual Pub and PRI's other work, and the other is the link for the rest of the schedule for the spring through the end of May. And thank you very much, Brandi. This was fascinating stuff.
Thank you for having me, and I've really appreciated being here.
Yeah, my absolute pleasure. And thanks to everyone for coming. And if there are other questions, you can shout them out. And otherwise, we'll call it a night.
Thank you, Brandi.
Thank you so
great.
much.
So interesting.
Yeah,
thank you.
Amazing stuff.
And I apologize. My 3-year-old is breaking down about now, ready for bed.
I don't hear her. I don't hear her.
Not hear that, but if you can go, then you.
She's collapsing outside the door as we speak.
Yeah, I'm collapsing too because I think the hydraulics in my chair has started leaking, so I'm dropping down, and so occasionally I pop back up. Pop back up.
Okay, take care.
Thank you so much.
Have a good night.
Thank you for your